sample and the associated, get rid of that workspace link shortcut. That are within the group that you have highlighted, and by default you have a statistic, which is, if you draw a gate, it'll be the percent of parent, and the number of cells that are displayed in the columns. But you can also add additional information in the form of keywords. You can either do this like I did before acquisition on the Diva setup, where I've entered a patient ID. In this case, these were humanized mice, so I have the mouse number, I have a time point, which is the day of collection, whether or not they've been infected with my virus, and then a numerical sort key, which helps after the fact, so it doesn't have to alphabetize things. I can add this information by going to the Workspace tab and adding keywords, or the Configure tab and editing columns. And I'm going to show you how to do that in just a minute, so I'll go over it again. When you're ready, you can double-click on a sample, and it'll open up a new graph window. And the graph window is where you can add your gates to identify subset populations of interest based on your marker expression. There are different kinds of plots that are available to display the flow data. To get to those plots within the graph window, you hit on the options drop-down, and then there's a drop-down menu here where you can switch from pseudo-color to various ways of displaying your data. So there's a 2D, a contour plot, a density plot, zebra, dot, or one-dimensional histograms. Really, it's whatever you like to look at, but I will say that I prefer to make gates on 2 dimensions if you're using a multi-dimensional analysis. If you are always gating on histograms, you're going to end up clipping populations that you could cut off more effectively in a 2-dimensional space. And then within that graph window, we have a whole bunch of gating tools up in the top left-hand corner. There's, I'm going to go through each of these as I go through. There's also an undo and redo button. You can go up and down the gating tree by using these up and down arrows. You can also move through the gating tree using the breadcrumb bar at the top here, and I'll show you how to do that. There is no right or wrong way to gate your data. It's really what best illustrates the analysis and best tool to subset your populations. And the tool that you use is going to vary on the distribution of your populations and your markers. And so don't be shy. You can always delete the gates when you make them. You can always change them, size and shape. So you have to kind of explore the tools and get a feel for them, and then decide what best fits your data and is appropriate for it. So let me go ahead and show you. In the program, 34 samples, FMOs, and all stain controls. I'm going to start with a no-stim LD1. That's just my background control. If I double-click on it, it brings up a graph window. I can make the graph window bigger if I like that. See it a little bit better. And then we can start actually drawing gates. All my tools are up here on the far left. One of the things I like to do is run all my assays in a plate, so a 96-well plate. And when I do that, I always make sure that there's no clogging or disruption of the sample over time. And I always add a time gate upstream of all my other analysis. And that way, if I do get a clog of DNA because of the cytotoxic killing in the stim or something like that, or a bad sample, which we all get in clinical cohorts, you can just move that time gate and cut off the bad sample. And usually side scatter shows the drop in pressure when you get a clog really effectively, so I can cut off bad sample and preserve at least some of my tube for analysis. So I start with a time gate. That's totally optional. And then I go to a forward scatter height versus forward scatter area. If your machine is set up, you have a nice diagonal between those two parameters, and what I'm doing here is going to take the cells on the diagonal, which are the singlets. Those are the single cells that pass the detector and are observable as single units. If it goes off the diagonal, I can't be sure that these aren't double cells, that there isn't a B cell and a T cell that pass by the detector at the same time. Those are going to show up as being CD19 and CD3 positive. They shouldn't exist, so I don't want to mess with those. I'll just take the 92% that are single cells. To move down in the gating tree, you notice as I make gates, it creates a hierarchy, and each subsequent node is indented to show that it is a child population of the parent. If I go up or down using the arrow buttons, I'll move through the gating tree. I can also use this breadcrumb bar, and I'll show you that. But if you double click on the gate, it brings you down to the next gating level. You can go back to side scatter and just do, maybe I'll take this ellipse tool as my lymphocyte gate, and just call those lymphocytes. FlowJo already knows it's got some favorite names that are in there. If you have something that you like to call your markers, if you hit this plus sign, it'll add it to your favorites and then it'll auto-fill in. You can also modify your favorite names in the preferences of the program. You can see the favorites that are existing right here in the drop-down menu. But then I just hit the arrow and it fills in the rest of the rest return. And so I have this nice big lymphocyte gate. I'm going to start my analysis by being more inclusive because I don't want to miss anything. And oftentimes when you stimulate cells, they'll clump up, they'll change in side scatter, getting more mature, more granular. And so I want to be more inclusive here. But I can also show you some options on this gate. First of all, the drop-down options, you can change the type of plot that's displayed. In this case, I like pseudo-color a lot for actually making the gates and drawing. Really easy to use. You can see the density, it's higher the red regions and lower with the blue. So you get distribution and density information, but I don't want to pay page charges for color publication of this plot, so I'm going to maybe go to a contour plot as an alternative. That doesn't look very similar, but if I click show outliers, then I can actually get a plot that gives me density information and looks pretty. So great way to publish. Other plots are great too. You got your dot plots, but it doesn't give you density information per se, and it downsamples to the number of events that you specify right here. And then we can always do a 1D histogram, but I like to stay away from those unless you're using them to illustrate a particular point, like a response of a functional marker. These are all my suggestions. There's no right or wrong way to do this. So go back to pseudo color, and then I want to show you some active gate options. So if I click on the gate, notice if it's grayed out, it's because I don't have anything selected. But if I click it, I can now gate events inside by default or outside. I can actually tint and show what I'm including in my gate. So now that's gated outside events, got gated inside events. I can make this gate magnetic, which will take the center of the gate and position it at the center of the most dense population there. And it'll show an associated vector of how it moved the gate. And so if you have lymphocyte drift in your samples over time, that might be a use of magnetic gates. Personally, I'm not a fan of them. I just like to go through everything by eye and make sure it looks good after the fact, but it could be useful. You can also increase the size of the gate or decrease the size of the gate right here with the plus and minus buttons. I'm going to go ahead and leave it as is there. Maybe I will make this one a contour plot, and then I'm going to double click, go down to the next gating level, and we can start actually looking at our fluorescent molecule.